The treatment of certain cancers, of infections such as HIV or invasive fungal diseases, or of other serious medical conditions, relies on the regular administration of critical drugs, which means drugs that are absolutely required to keep those life-threatening diseases under control. Quite a number of these critical drugs are candidates for the monitoring of their blood concentration in the patients who receive them. Indeed, the concentration monitoring allows a precise personalization of treatments. This can improve long-term patient care, in particular for drugs with a highly variable and poorly predictable relationship between dose and concentration among individuals. This is particularly the case for drugs having a narrow therapeutic index and lacking clinically relevant biomarkers of efficacy and tolerability. And the aim of the IsyPem project is precisely to develop a new approach for therapeutic drug concentration monitoring at the point of care. At present, concentration measurements are performed in large medical laboratories using modern technologies, such as fluorescence polarization or liquid chromatography with mass spectrometry. The measurement results are sent to medical doctors in charge of the patients. However, the clinical interpretation of results represents a true challenge for non-expert practitioners. Indeed, it is fairly difficult to translate a mere drug concentration value into an appropriate decision of dosage adjustment in the patient. Clinical pharmacologists are experts in the field, but they are not largely available.
For monitoring to be useful, it's important that it appropriately informs about dosage adjustment decisions. And this requires that answers are brought to three main questions. First, whether the result is normal. This means within the range expected from population data considering the current dosage, the time after the last dose intake, the time of blood sampling and various patient-specific factors known to affect drug disposition. Second, whether the result is in the target range appropriate for the patient considering available evidence from clinical studies which have described the relationships between concentration, exposure and both efficacy and toxicity. And third, how the observed concentration value predicts the future exposure that shall result from a given dosage adjustment, so to lead the patient's blood level into the suitable target range.
This is exactly what the IsyPem software aims to do, to assist physicians in the clinical interpretation of drug concentration results. The concentration value is entered either manually or through the direct connection with a lab system or an electronic medical record. The normality is evaluated by comparing the observed concentration value to a family of curves representing the probability percentiles expected under treatment. This evaluation is based on population pharmacokinetic parameters that are plugged into the program as an editable set of data. This set of drug-specific parameters also informs the user about target ranges that are usually associated with good treatment efficacy and tolerability. Finally, the software calculates a maximum likelihood concentration curve for the patient, derived from both his or her concentration value and the population pharmacokinetic parameters through so-called Bayesian calculation. It then proposes several scenarios of dosage adjustment and predicts the level of exposure expected under each scenario. Once the physician has decided upon dosage adjustment, the program issues a printable report. The program will also record all utilization data into a local database with possibility to collect them centrally in order to help further research.
This software will accommodate parameters for a wide range of drugs. It's supposed to be disseminated largely about a large community of users who are ready to bring further drug parameters and to share their observations and their studies throughout the community.
And more importantly, the IsyPem application is meant to be integrated with a point-of-care analytical system for the measurement of drug concentration in blood. This is also a part of the IsyPem project. Ultimately, the aim of the project is to provide the concentration results to the prescribers and to be immediately exploited in order to support the dosage optimization of critical drugs.